inviting me for this event. I'm really excited to talk to all the teachers on the call and those who are also probably watching the video afterwards. So I'll start by sharing my screen. Let me see if I can do that. So I'm an assistant professor in the physics department at Virginia Tech. Today, I'm going to talk to you about one aspect of my research, which I thought would be very beneficial for your students in terms of kind of broadening their perspective about what STEM is. Many students have a perspective of STEM as a field that they would go into if they're interested in academia or industry. But today I'm going to tell you about another opportunity, which is going into national labs. My background is in neutron scattering, so I thought that I would give you some a feel of how we use neutrons, how we play hide and seek with neutrons in order to study matter on the nanoscale. And this is what I do in my lab. Comment about myself, and the reason I'm mentioning this is to kind of tell you that science is flexible and career trajectories are not necessarily straightforward. And you'll see in a bit how I actually have changed my field of research in a way that adapts more to my strengths, which is exactly what I'm currently doing. All right. So I currently work on soft matter systems, soft matter physics in particular. I'm interested in how molecules assemble and arrange and move in order to provide or to give us materials with certain performances. But the way that I started my career is back home. I'm Lebanese, so I come from Lebanon. I did my bachelor's degree and my master's degree back home. My master's degree was in supersymmetric gauge theory, so this is quite far from where I am right now. I kind of joke to my friends sometimes that I've been going down the energy scale, but this is kind of just to give you a perspective about how flexible things can be in science. So even if your students start somewhat and they don't necessarily like it, they can actually change their fields of research or fields of interest, and that shouldn't affect their career trajectory. All right, so after I got my master's degree at the American University of Beirut, I moved to Indiana University in Bloomington, where I did a PhD in neutron scattering applications on periodic nanostructures. The reason that I like these photos is to give you a contrast between the sunny Beirut and the really snowy Bloomington, Indiana, which was a little bit of a shock to me, but I managed to survive okay during my 5 years of PhD. So after my PhD, I actually moved around quite a bit. I started here at Indiana, and then I went to the National Institute of Standards and Technology, where I did my postdoc in polymer nanocomposites and lipid membranes. Again, this is a change in the research area, just because I was interested in learning more and broadening my perspective of science in a sense. After that, I moved to Oak Ridge National Lab, where I did a Schill Fellowship using neutron spectroscopy to study and understand lipid biomembranes, which I'll be talking to you a little bit about today. And then after about a year and a half, I moved to Virginia Tech, where I started my faculty career about 2.5 years ago. All right, so if you can see from the topics of research that things can change over time, and this evolvement of interest is not necessarily a bad thing in science, especially nowadays where there's a lot of interdisciplinary research going on. All right, so before I start, I thought I'd share with you some advice that I tend to give out to both students and teachers whenever I can. And that is basically to kind of assist you in a way to convey relevant information to your students. And I'm happy to answer any questions about this later on. But I thought that I should mention that many students have weaknesses. And one of the roles of teachers is to encourage students to figure out their strengths and their weakness and to choose their career trajectory accordingly. And I give an example about myself when I started in high energy physics and I moved to Bloomington, Indiana, where I joined the high energy group in the beginning. I thought this was a nice application or a nice extension to my master's thesis. But I soon realized that these projects take sometimes up to 10 to 15 years to materialize or to give results. And one of my weaknesses is that I am not that patient to see my results. Okay, so I thought that this is a career trajectory that will not work for me. So I actually switched into condensed matter physics after my first year of graduate school, and that worked really well in my favor. So this is an example of where realizing strengths and weaknesses can be extremely helpful in figuring out one's career trajectory. The other thing that I do want to share with you, and I actually serve on many boards that are advocating for inclusion and diversity in science and engineering, in STEM in general, is that women and minorities can feel large barriers in pursuing STEM fields. So as teachers, you have a lot of influence in trying to help your students overcome these barriers. And a lot of them are actually driven by stereotypes and lack of immediate support networks, whether it's family or immediate environment. So teachers can play an extremely important role in making students realize that nothing is impossible. The other thing that I do want to encourage or ask you to do is to encourage your students to engage in professional development activities. I'm going to give you examples about things that are happening in physics. For example, the American Physical Society hosts a yearly conference, an annual conference that is the Conference of Undergraduate Women in Physics. I know that the name says undergraduate, but we actually accept a lot of applications from high school students, primarily women, who are interested in pursuing physics and are uncertain about how to go about it. Many students also tend not to seek advice. So you can encourage your students, to seek guidance and also inform them about possible research opportunities in university labs. One thing that I do want to mention there is that most faculty are actually quite interested in hiring or mentoring high school students. Many funding agencies ask us to do this, and this is kind of a mutual interest. We are interested in mentoring high school students, and I'm pretty sure that many high students are also interested in gaining that research opportunity early on in their science careers. So I think that this is something that extremely important to point out. One of the things that Kim asked me to talk a little bit about is what is needed to succeed in STEM. And in my opinion, and in my field of research, I thought that these 6 bullet points are extremely important for a student to succeed in STEM. Particularly in physics, there's mathematics play an important role. So students need to have a good mathematical background. They also need to have a general curiosity for learning and exploration. And they should recognize that even if they have knowledge gaps, they should be willing to overcome them by working a little bit harder on what they lack in terms of clarity of concepts and such. Students also tend to have this glorified perception of what success is. And I think that success is kind of a straight line or a linear trajectory where if you do something, you'll eventually succeed, or you will succeed. But in fact, success can be a random walk where you can have many failures until you eventually succeed. So realizing that success can come after failure is really important in kind of having a clear perspective of what it takes to go into science. There's a lot of persistence and dedication and work that goes into having a project succeed or succeeding as a student in STEM. And nowadays, with merging of fields, I think that students should realize that it's important for them to work smart, because a lot of that depends on kind of making connections between fields that is really, really important. And it kind of just takes a general perspective of what a student wants in order to achieve or to succeed in a STEM field. I also would like to point out a few student opportunities for research. Again, I want to emphasize some aspects that other speakers might not talk about. As I mentioned earlier, there's always research opportunities in university STEM labs. I myself am highly interested in mentoring high school students for research opportunities, because this is something that my funding agencies are interested in, and this is something that I am interested in terms of educating the next generation of STEM workers in science skills and advanced skills that are needed for national needs. There's also summer internship in national facilities. This is something that not a lot of people know about. I'm going to give you a couple of examples from NIST, which is the National Institute of Standards and Technology, and ORNL, which is Oak Ridge National Lab. So NIST has this summer high school internship program where high school students can apply to actually spend a couple of months at NIST, where they work directly with scientists there in order to work on a given research project. And I think these opportunities are extremely useful in terms of applying to STEM fields for college degrees. Oak Ridge has a similar program that is called the High School Research Experience Opportunity, HSRE. And there are other opportunities in other national labs. I'm just kind of giving you a flavor of what this is in labs that I'm familiar with. The other thing that is kind of indirectly goes back to students is research opportunities for teachers at university labs. I myself am interested in working with students over summer to pass on some of the expertise or skills that they can eventually teach to their students. And in that case, teachers can be the facilitators for their students in pursuing research opportunities. Again, this exists both in university labs as well as in national facilities. The other thing that you can consider is inviting research active faculty to talk to your students about the research and internship opportunities in the labs. And this is, again, this is a mutual interest. And of course, with STEM becoming more interdisciplinary, this offers remarkable flexibility in terms of pursuing research interests or STEM interests, and also finding career opportunities that match the acquired skill sets that students get during their STEM degrees. All right, so with that, I just kind of want to briefly reiterate that there are more than just academic and teaching opportunities for students going in STEM. There's also industry, pharma, national facilities, and among national facilities, there's NASA and other national labs, including NIST and Oak Ridge. And national facilities actually offer a very interesting career environment that is somewhat intermediate between academia and industry. So this might be of interest to students who are not necessarily super excited about either academia or industry, but are still interested in STEM fields. All right, so with that, I will move on to tell you a little bit about what I work on. So I work in soft matter physics. And what is exactly soft matter? This is everything that is squishy, from the cells in our bodies to the tires in our cars to toothpaste, soaps, gels, and liquid crystals in our computers. But of course, I don't study all of these materials. What I'm really interested in are two types of materials, which are polymers. Those are plastics that are enforced with some particles. And then, of course, I work with biomembranes, which I'll tell you a little bit about today. All right, so why are soft materials so interesting? Well, they're so interesting because they are squishy. They actually exhibit a rich space of structure and dynamics. And because they're squishy, because particles in soft materials, unlike solids like magnets or crystals, which are usually bound by really strong forces, in soft materials, particles and molecules are bound together by very weak forces, which allow them to move around rather easily. And this is why these structures evolve and also the motions evolve over time. So this is why these materials can actually change their properties over time. And one of the things that is of utmost importance at the moment is trying to understand how these properties evolve with aging or over different time scales. The other thing about these materials is that because they're so squishy and because the forces between them are quite weak, they actually have very small energies in very short relaxation rates. And so this makes them really hard to study. I mean, if you think about very large objects like the planets or even objects around us, we can study them on reasonable time scales like seconds or hours or even years. What I'm talking about here in these soft materials are motions on the order of nanoseconds or picoseconds or femtoseconds. So these are extremely small length scales and time scales that require us to use sophisticated techniques other than macroscopic techniques that we are used to in understanding the world around us. So one of the things that I'm really interested in applying these sophisticated methods on the systems that I'm interested in is in a sense to understand the machinery of life, to understand how nature has evolved over the years in order to give us very delicate biology that is, and also materials that are governed by very precise motions and structures that give us the properties of matter that we're interested in. Biology in particular offers a great deal of precision and selectiveness that we're still trying to understand and emulate in engineering materials with similar properties. So because these systems are very complicated, of course, to understand all of the parts that are involved in the function of all of these materials, it becomes really important to understand what each of the elements or each of the parts of this complex machinery does, okay? And this is where neutron scattering and scattering in particular, or in general, becomes really effective because we can start to apply interesting physical laws and interesting physical phenomena in order to selectively study parts of a complex system without affecting the other parts. And the way we do this is by using a version or an analog of optical contrast, which I will get to in a minute. But if you can think of this complex gear system, let's assume that we are going to simplify it into 3 gears that are color-coded. Now, if you can study this system in an environment that matches out the red gear, for example, this allows us to understand what the black and the gray gear are doing. So this is just a schematic or a pictorial depiction of how we can start to understand how the parts of a complex system work together, work in unison in order to produce function. And the way that I do this in my lab is using neutron scattering methods. All right. All right, so how does neutron scattering work? Well, I usually give an optical analog, which many of us know and realize because we see this every day around us. And a clear example of this is what we see here. We have a beaker in the right, which has water, a beaker in the left, which has vegetable oil. If you put the Pyrex rod in each of these beakers, you will be able to see the Pyrex rod in water, but you won't be able to see it in oil. And that is mainly because the index of refraction of Pyrex matches that of oil, which makes the rod invisible in Pyrex. We play actually similar rules or similar games to this on a molecular scale in our measurements by using the, you can think of it as the index of refraction that neutrons see in order to study material differently. One thing that is so particular about neutrons is that they're isotope sensitive. They're sensitive to the nuclear structure of matter. And so what we can do there is we can replace hydrogen, which is so abundant in natural materials and in biological systems, by deuterium, which is exactly the same atom except additional neutron in its nucleus, which makes the index of refraction that is seen by neutrons completely different on a molecular scale. So you can think of this example of a Pyrex rod in water and in vegetable oil on a molecular level, happening on a molecular level, which allows us to study how molecules and collective behavior occurs on nanoscales by applying these optical tricks. All right. So what exactly are we interested in? Well, I am not a biologist. I'm actually a physicist with interest in biophysics. My lab is primarily interested in how the assembly of lipid molecules and their motions result in specific biological function. So we're studying this from the point of view of biophysics and physical assembly. And what we're really interested in is trying to understand how lipid molecules in a lipid membrane, which is usually a model system for our cell membranes, work together in order to produce function. Again, we use neutron scattering methods. And I give you here an example of a schematic of a lipid membrane with a protein in it, where we can match the environment by changing, let's say, water into heavy water, so H2O into D2O, by just simply changing the isotope labeling of the system to either focus on the protein, as you see in this middle graph here, or to focus on the lipid membrane by completely masking out the protein, which now allows us to start understanding how lipid membranes interact with proteins, for example, on a molecular level and on the nanoscale in order to create function. Okay. So just to give you an idea about why exactly we are interested in biomembranes. Well, we're interested in them because they're actually quite exciting model systems for self-assembly. The way that our cell membranes are formed are they're primarily formed of what we call phospholipids. These are lipid molecules that have a hydrophilic head that likes water, and a hydrophobic tail region, or two hydrophobic tails that dislike water. So when you put them in a water environment, what happens is that these lipid molecules basically zip on each other and form what we call a lipid bilayer. And this is a way to shield the tails from water, in which the heads are now facing water, and they minimize the energy that is required for the system to exist. So this is a classic example of self-assembly. We're just putting molecules in a given environment, given the forces that are required in that system. Molecules start to self-assemble in really exciting properties and really exciting structures, such as the ones that we have in our cell membranes. But cell membranes are much more complex than that. Even in the same cell membrane, you can actually have tons of lipid species. What we're really interested in are just two kinds, phospholipids and cholesterol. And even with that, these can come in a variety of flavors, depending on the chemistries of their tails, whether that has double bonds or not. But it also, these structures also tell us about the properties of these cell membranes. So I like to give this slide where I tell people that we are what we eat. And basically it's because the foods that we eat, the fatty acids in the food that we eat are very similar to the chains of the lipid molecules that form our lipid membranes. What we're really interested in here is figuring out how these different chemistries result in different physical properties and eventually in function, okay? And I think I'm running quite slow, so I'm going to try to speed things up a little bit. What we're really interested in is understanding particularly how the dynamics between lipid membranes and proteins work. And that is because both membranes and proteins are highly dynamic. Membranes can exert or exhibit different modes of fluctuations or deformations, whether it's bending or stretching or thickness changes. My lab is primarily interested in these two modes, bending and thickness fluctuations, and we study that with neutron spectroscopy. So the way to understand these lipid membranes is we actually, again, I'm a physicist, so I work with model systems, and what we do is we take lipid molecules, put them in water, and then extrude them through a polycarbonate filter to form these fascicular structures, and then this allows us to study them in different modalities. This is an example of what we can see if we use neutron scattering on lipid vesicles. This is an example of how modeling works with scattering techniques. And I'm mentioning this in particular because really nice learning opportunities for students in terms of modeling data. We also use spectroscopy methods where we look at the fluctuations of these membranes on the nanoscale. Actually, if you see the time scale here, these are, this is in nanoseconds. And of course, we can apply different isotope labeling methods to understand the different fluctuation modes in these membranes. If you look just at bending fluctuations, you can get physical quantities of membranes like bending rigidities. You can also do isotope labeling by matching the tails of the lipids to the water environment around them, and this allows us to get another biophysical property of membranes, which is viscosity. And both of these properties are really important for function. All right, so I see that I still have about 15 minutes left, so I'm going to try to wrap this up. So again, I'm showing this slide because we work with model systems. The easiest model system to work with to emulate what our cell membranes looks like is basically by taking a single component lipid bilayer. And we've studied different types of single component lipid bilayers, which vary in chain lengths or chain chemistry. And we understand the phase behavior by simply applying density measurements, which our students should be very familiar with. In fact, I have a densitometer in my lab that high school students can use in order to understand physical phenomena and phase behavior on a molecular level by looking at these macroscopic properties of lipid membranes. We can also look at how a change or a transition in the phase between a gel phase or a liquid phase in membranes can affect their physical properties. And of course, one thing that we do in my lab is to use also simulations to provide insights into molecular behavior. So this is an example of how simulations match both the structural and the dynamic signatures that we see in our experiments. So I'm showing you here an example of a slightly more complicated system where you have now two types of molecules. So those are binary membranes instead of single component membranes. And you can start seeing how just including two types of lipid molecules, we start to get features in our model systems that resemble what we see in cells. These domains here are what is usually referred to as lipid rafts, and they're thought to be platforms for protein recruitment and cell function, right? So we're studying these from very basic principles, very basic physical principles to try to understand how cell membranes evolve and how cell membranes work. Another cool example that we're currently working on is this understanding how membranes interact with different types of proteins. This really cool type of peptide that we're currently working with that is called the pH low insertion peptide, which binds to cell membranes at the neutral pH, which is what we have usually in healthy tissues. And it inserts itself across the membrane at acidic conditions. And using neutron spectroscopy methods, we're able to see that the insertion of this peptide suppresses the fluctuations of the membrane, which we also validated with simulations. And this is basically, we've discovered a new mechanism by which membranes and proteins can interact to provide signaling pathways. We're also interested in how membrane functions affect their, or membrane curvature affect their functions. And we do this by borrowing some ideas from nature. Nature tells us that curvature plays an important role in moving these lipid domains that I just showed you earlier into more curved regions. And in fact, the way that we do this is using what we call smart materials or thermoresponsive or stimuli-responsive polymers, which allow us to change topography to understand how function works. So I'm not going to go into too much detail in there, but this is starting to become a combination of engineering, chemistry, and physics. And this is just to give you an idea of the transdisciplinary nature of research that we do. I also have some lab capabilities that are more appropriate to high school level. Those include Langmuir trough capabilities and differential scanning calorimetries, which allow us to understand membranes on a molecular level, but using macroscopic observations. And I've had students work on both. All right, so I also work on polymer nanocomposites. I'm not going to go into the details of that project, but I do want to finish with a video of what goes on in a neutron scattering experiment and how we can utilize that to understand matter on the nanoscale. So I'm going to try to see if this video works. I think I have to get out of laser pointer mode. And let's see if this works.
What matters most to a researcher doing neutron scattering?
All right, so I'm going to stop here. I just wanted to share with you that these are some of the capabilities that I have in my lab. And in fact, high school students have the opportunity to engage in any portion of these experiments, including reducing data or looking at raw data and also analyzing data from these very highly advanced resources that one can find at national facilities. So with this, I will end my presentation. I'd like to thank you all for your attention, and I'll be happy to take any questions.
Oh, Rana, thank you so much. I always, I travel and see a lot of presentations, and I think my favorite ones are always the ones that are so interdisciplinary and are really borrowing from a lot of different fields. And I think especially it's so illustrative of the connections that various science and engineering branches have with each other, certainly. So a couple of questions.
Yes.
So first question is, how do you help students discern the difference between strengths and weaknesses and understanding that in a given field there will be challenges, and just because something is difficult for you, it is not necessarily a weakness?
That's actually a very good question. And it really goes back to whether the student is willing to take on additional work to fill these knowledge gaps that they have. Absolutely, if something is difficult, that doesn't necessarily mean that it's a weakness. What I was mostly referring to in the beginning as a weakness is nothing is a weakness. Anything can be changed into a strength, right? But also knowing what you want and what you are interested in is important to realize what you can do on a long term. So I just gave an example about myself that I don't have patience to see my results come in after 10 years of work. I think of that as a slight weakness, but it's also something that I can bring to my advantage. What I did there is I changed fields in order to address that issue that it's not something that I can change overnight. So students have to realize what is it that they're able to work on, what is it that they're able to overcome, rather than just give up and say, this is not something that I can do.
Oh, no, thank you. And I think I still struggle with that. And it's something I think about all the time.
We all do.
But I do think your example is great, because I too don't like to wait for things. So that would also not be for me. Probably why I enjoyed.
Yeah, for someone who, for example, does not work okay with kids, for example, they shouldn't go into a teaching career. For someone who's not really interested in putting long hours in the lab, they shouldn't go into a research-focused career that requires a lot of lab hours. So it all depends on what students are interested in and what their aspirations for a career are like.
That's why I didn't end up in a lab. Turns out I do not like bench work. So another question, my honors chemistry students carry out a year-long project where they choose a topic, decide what to test, and carry out research and an experiment, though not always terribly complex. Do you have any suggestions to help make sure the experience is developing the skills that will help them move forward in their field of science?
So my question there is this happening at a school level? Or is it happening is this honor student doing this research in a lab, in a university lab?
No, I believe I assume high school. Julie, you can just chat in and correct me if I'm wrong. So
it is in a school level.
Yes.
Okay. So I think that any research experience is a good experience as long as the student is aware of debugging what they're getting. For example, debugging is a huge issue in research. Sometimes you get results which make or do not make sense. And one way to figure this out is to figure out, for example, are our experimental settings the way that we expected? What is going on in our experiment that is giving us this feature that we cannot explain? So any research experience can develop good skills as long as it is directed appropriately. So even if this research is happening at the school level, the teachers can contribute a lot in terms of guiding the students into figuring out how to interpret their data and how to think of experimental settings that they're working with. I don't know, Julie, if this answers your question. If not, feel free to reach out to me by email later. We can also set up a meeting if you're interested.
Yeah, and everyone will get our speakers as well as mine. Most of you have my contact information, but myself and Molly's contact information as well. So one more question.
Sure.
So you, coming to this from a physics angle, I guess curious, and I'm curious to know, you ended up in this sort of biophysics field, and I guess what I would like to know is, like, where'd you get your biology knowledge?
So here's a disclaimer. I hated biology in high school. I actually did not take biology in high school. So this is what I'm saying. Things evolve, right? I hated biology in high school because in middle school, biology was a set of memorizing names, and I'm not good at it. I'm not good at memorizing. I'm good at making connections logically, but I'm not good at memorizing names. So I opted, we have a system back home where you can opt to take math and physics focused curriculum in high school, or biology and chemistry focused curriculum. So I opted for math and physics, and I developed my interest in biology in graduate school. And I'm approaching this from a fresh perspective, and this is what I think is an advantage, because I'm looking at these problems from a fresh perspective, where I'm bringing in the knowledge from physics and seeing how it applies to biology. So this is now a super exciting time to be in science, because fields are merging, because we have a lot of transdisciplinary projects and research opportunities that people can contribute to from different perspectives. And this is what drives science forward.
Do you work with biologists?
I work with biologists, but from a very model point of view. So we work with model systems. I don't work with actual cells. I actually work with model systems that mimic cell membranes, and I try to understand them from a physics perspective. But you are absolutely right. Working with biologists has been extremely tremendous. I work with biologists, but we try to kind of find a connection, find common grounds, and this is what is enabling my research to make impact, because where biologists understand things from a biology perspective, I'm approaching this from a physics perspective and finding these very exciting phenomena that are happening on the nanoscale, which we had no idea about before now.
Wonderful, that's so exciting.
Yeah, thank you.
So thank you so much for presenting. Normally everyone would clap now. But thank you so much.